Okay, so we're about to start.
Hello everyone, welcome to today's live broadcast on risks of bias in vivo research and what to do about it. My name is Emily Senna from the University of Edinburgh and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Professor Malcolm MacLeod. Malcolm is a professor of neurology and translational neuroscience, whom I have had the joy of working with for the last 11 years. Malcolm's research interests revolve around developing tools to provide unbiased summaries of what is already known, including tools to assess whether effects in animals may be overstated, and then using this information to help guide better design of clinical trials testing treatments in humans, sorry. I'll now turn it over to Malcolm for his presentation.
Well, thank you very much, Emily, and thank you everyone for dialing in. What for some people will be quite early in the day and for others will be quite late in the day. So over the next 50 minutes or so I want to give you a whistle-stop tour about what I think are the important risks of bias in animal research and what we as research practitioners might do about it. I should start off with some disclosures. I'm a member of the UK Home Office Animals and Science Committee, responsible for setting at least some of the regulatory framework for animal research in the UK. I'm a member of the UK Commission for Human Medicines, which is responsible for giving regulatory recommendations for approval for new medicines. But most importantly for you, I have sought in the past and will continue to seek funding for work in this area. So you will understand if I try and tell you that everything that I do is fantastic and excellent and very, very interesting. And of course, people should give me lots of research funding to do more of it. So I'm a translational neuroscientist. Here's an example of a translational problem, and we have quite a few of these signs both in Wales, where this is from, and in Scotland, where road signs are given in the English language, and then underneath there's a translation of what it means in the local language, whatever that might be. And this sign says no entry for heavy goods vehicles, residential site only, but in fact the Welsh translation says I am not in the office at the moment, send any work to be translated. So this translational road signist, if you like, has misunderstood the black box into which they have sent their data, their English text string, and accepted without any further thought what's come out of their black box machine. In this case, one assumes an automated email response. And some of the problems that we have with research come about for these reasons. Here's another example. This is work using a technique called functional MRI scanning. The premise of this is that when part of the brain is active, it requires more blood. This is achieved through a vasodilatory process, increasing the local blood flow, and that change in oxygen level in the blood can be detected in a functional MRI scanner through something called the blood oxygen level dependent signal. Now these machines are very temperamental. They often require to be set up very precisely to have their knobs twiddled and their egos tended so they give the right pictures. And this group in Boston used to set up and trim their machine every Monday morning, and they had a competition to see who could bring in the most outrageous piece of organic material for the setup of the scanner. And one morning it was Craig's turn, and on his way to work he remembered that he'd forgotten to bring in something organic for the scanner. But fortunately his route to work took him past the Boston Fish Market, And he realized that no one had had a salmon in the scanner before, so he got a salmon. And he took it into work, and they set up the scanner, and they twiddled the knobs until the scanner was working properly. And then their human volunteer for that morning didn't show. And the genius that he had was to put the salmon through the fMRI scan. So one mature Atlantic salmon, weighing about 3.8 pounds and measuring 18 inches long, participated in the test. And the salmon was asked to perform a task. The task involved picturing a series of photographs depicting human individuals in social situations where there was a specified emotional valence. So the human was happy or sad or perplexed or confused, and the salmon was asked to determine which emotion the human in the picture must have been experiencing. Several active voxels were found in the salmon's brain cavity, close to its equivalent of the human orbitofrontal cortex, which lights up in most of these scans, at a very high significance threshold. And as they say, either they've stumbled onto a rather amazing discovery in terms of post-mortem ichthyological cognition, that's to say that their dead salmon could think, or there must be something wrong with this methodology, with this approach that gave them what must be a false positive result, for which they were awarded the Ig Nobel Prize in Neuroscience in 2012, and rightly so. And closer to home, this is work from Ruth Dayton, who's an Edinburgh neuroscientist, unfortunately not in my group, but she did something very smart. She was interested in looking at cell death in a cell line called the SHSY5Y cell line. So she exposed these cells to staurosporin and then looked at changes in messenger RNA expression between the ground unstimulated state and the dying cells to see which genes were turned on in this process of active cell death, of apoptosis induced by staurosporin. And she got 13 genes that were regulated, and she sent them away to a website called Ingenuity Pathway Analysis that comes back and tells you if the genes are regulated together in a pathway, and if they are, what that pathway is. What it does is give you an IPA score, and the IPA score is the negative logarithm of the odds of those genes being associated together by chance alone. So an IPA score of 10 would mean that the p-value was 10 to the minus 10. So she got her results back, but they didn't make any sense to her. The machine seemed to be telling her that these genes were regulated in a way which she thought was implausible. And rather than believe the machine, our Welsh road scientist's problem, she thought she'd test the machine. So she went to the gene base and selected 13 genes at random and sent them to IPA. And then she did it again and again and again, and she did it 1,000 times with 1,000 sets of 15 randomly selected genes and got back the IPA score. And here's the distribution of the IPA score in this graph on the right, and you can see that the IPA scores range from 4 to 38. So it didn't call any of these random collections of genes at less than 1 in 10,000, and the median score was 1 in 1 million. So what this tells you is that genes are regulated together by chance alone because they happen to be in the same cells serving the functions of that cell. But these kind of web-based analysis tools often don't tell you very much more than the bleeding obvious. And here's an example much closer to home for me. This is testing a drug in an animal ischemia model. This is a model of ischemic stroke in the rat, where the rat is exposed to either control conditions, and you can see an infarct volume of about 200 cubic millimeters, or half-dose low-dose glutamate, and then full-dose low-dose glutamate. And you can see a reduction in infarct size in association with this drug treatment. And I've not shown you the statistical significance testing, but it's statistically significant. But the problem is when I tell you that half-dose low-dose glutamate is 10 to the minus 120 molar, and full-dose low-dose glutamate is 10 to the minus 60 molar, then those of you who are familiar with Avogadro's constant will know that there isn't any glutamate in either of those columns, and so there must be something wrong. Well, either homeopathy works, and I don't think it does, or there's something wrong with this widely used experimental technique or the way it's analysed. So we were interested in translating treatments from laboratory models into clinical trials in humans, and over 10 years ago identified 1,026 drugs which had been tested in some form of in vitro or in vivo stroke model. Over 600 tested in animal models of focal cerebral ischemia, of which 374 remarkably improved outcome in those animal models. So one would imagine for a translational stroke neuroscientist like myself, there'd be a great deal of work for us to do. And in fact, some of those drugs had been tested in clinical trial, 97 of them. And of those, one worked in human stroke. That was clot busting treatment with TPA. Interestingly, it was tested in human stroke, not because of the animal data, although there is animal data, and Emily, who introduced me, has systematically reviewed those animal data. But it was tested in human stroke because it was affected in a cognate human condition, myocardial ischemia, myocardial infarction. And in fact, there are 3 treatments which are in routine clinical use: aspirin, stroke unit care, and decompressive hemicraniectomy, all tested in human clinical trial without ever being near an animal. So there's a real question as to the extent to which laboratory research, animal research in stroke has contributed so far to the development of effective treatments. And what I'm interested in is why that might be. And I think that preclinical trials fail to translate if animal studies mislead, and animal studies will mislead if their conclusions are confounded by bias because of how the experiment was done, how the data were analysed, if incomplete data are available to the people making the judgments about proceeding, if the studies used the wrong sample size or if they used the wrong model. And I'll try and cover some of those issues as we go through. So here's the story of a drug called NXY059, developed by AstraZeneca to treat focal cerebral ischemia, to treat stroke, and test marketed under the trade name Ceravive, so certain they were that they would make one's cerebrum survive following a stroke. They arranged in total 3 clinical trials involving 5,500 patients, and in the 48 hours following publication of the last, again neutral clinical trial, their share price fell by 17%, a market valuation of $9.6 billion, and it took them 6 years to get that market valuation back. So a disaster for them. And when we looked after the event in a post-hoc systematic review at the animal data which had supported the clinical trial program, we found high headline efficacy with a 44% improvement in outcome, but not very much animal data, fewer than 600 animals in total, and efficacy much reduced in high-quality studies, and I'll show you that, and much reduced in animals with hypertension, and I'll show you that also. So what toolbox do we use? Well, we use systematic review and meta-analysis. There's a number of excellent review articles about these, but systematic reviews are a review where the criteria for identifying and considering information and data are determined in advance and are transparent. And this contrasts with and is much less biased than narrative review. And meta-analysis is a statistical technique to combine data from separate experiments. You can use it, and clinicians often use it, to give an overall best estimate of a treatment effect. But we use it more to understand the impact of other things on what that treatment effect is. And those might be related to the treatment itself, the dose, the timing. It might be related to the animal, the age of the animal, its sex, whether there's any comorbidities. And importantly and crucially, it might be related to the design of the experiments. And this and a number of other issues in research are set out in a series of articles in The Lancet in 2014, published almost exactly 2 years ago, which identify 5 key stages where research value can be increased and research waste might be reduced. And I'm going to discuss in a bit of detail as relates to animal research that studies might take adequate steps to reduce bias, that studies are published in full, and that we report studies with disappointing results. But I recommend that series to you as a primer of what can go wrong, because as you know, what can go wrong usually does. And so the first thing is how the experiment was done, and I'd like to take you back to the 1960s to Robert Rosenthal and colleagues who used to teach a course in graduate psychology. And at the end of the course, their students had to perform an experiment. And the experiment ran over a week, and they were comparing cognitive performance in 2 cohorts of animals. One had been bred over many generations at Berkeley, then a leading center for cognition research. They'd been bred to perform well in cognitive tasks such as the T-maze, although not the T-maze itself. And the other group were maze dull. They're slightly thicker, if you like, country cousins who'd been bred and inbred over many generations to perform badly. And the task administered to the animal in the T-maze was placed in the stem of the maze. One of the arms of the maze was always bright and the other was always dark. which was dark and which was bright was flipped according to a predetermined random schedule, and the dark arm was always reinforced with food. So rather like the junior postdoc, the rat was being trained to turn to the dark side. And what they found was, firstly, that the maize bright animals performed better even on the first day of the experiment when they were naive to the cognitive test. And while both cohorts of animals improved over the 5 days, the improvement was much more pronounced in the maize bright animals than the maize dull animals. Furthermore, when he asked the students what they thought of their animals, the maize bright animals were inquisitive, friendly animals, the sort of animal you'd like to take home to meet your mother, whereas the maize dull animals were surly and insolent, and in fact 3 of the students were bitten by their maize dull animals. And in fact the experiment that Rosenthal had done wasn't on the animals, it was on the students, because those animals had been picked at random from the same cages in the same animal house on the first day of the experiment, and the only difference between them was in the minds of the students doing the experiments and the way those students handled the animals during the experiment, presumably induced by their precognitions, their preconceptions about what the animals were like and how they would perform, and perhaps that's why they got bitten. So going back to NHY-059, here's risk of bias in those studies, looking at infarct volume as an outcome, 408 animals. Again, we see this overall improvement in outcome in the dark grey bar, but when we look at studies that are randomized, which blind the conduct of the experiment, which blind the assessment of outcome, then we see there are substantially lower efficacies, significantly lower efficacies in those animals at low risk of bias. Not one of those 11 publications met all 3 of those risk of bias items, and the 2 that had been used by Astra to fuel their clinical trial program met none. So, with the benefit of hindsight, the reason that NHY-059 did not work in human stroke is probably because it doesn't work in animal stroke if the experiments are done to a high enough quality. So when we produced these data some years ago now, we were told that that was because stroke animal researchers were lousy. They didn't know what they were doing, they didn't know how to do proper experiments. We needed to look at proper neuroscientists because they would know what was happening. So we looked at some other neuroscience domains. Here's blinding again, this is from stroke. And then experiments in Alzheimer's disease and the Morris water maze testing the efficacy of candidate drugs in transgenic models of Alzheimer's disease, Kieran Egan's work, Hannah Vesrainen's work in multiple sclerosis, and Evie Rock's work in Parkinson's disease, showing consistently that there is significantly lower estimates of efficacy when the investigator is blinded to treatment group allocation. So that's where we are there, and then there are problems with the way that the studies were reported. So if, for instance, if we look at Hannah's systematic review of studies in multiple sclerosis, we found almost 10,000 publications testing about 1,700 interventions. We were interested in about 40 interventions that had been tested on 5 or more occasions and trying to extract the data from those publications for those drugs. And what we found that of 202 potentially includable publications, we had to exclude fully a third of them, either because they had no valid outcomes, no errors, didn't report the number of animals per group, didn't report the variance or the number of animals per group. 3 were completely uninterpretable, and 2, in spite of being experiments with a control group, didn't provide any outcome data for animals in that control group. So very difficult to understand what they actually did and to understand what's happening. And looking across a range of disease model areas to see where there is just problems with neuroscientists. We've now looked across Alzheimer's disease, focal ischemia, glioma, Huntington's, hemorrhage, multiple sclerosis, myocardial infarction, and spinal cord injury. Over 2,671 publications identified in the context of systematic reviews which we've performed. And you can see reporting of randomization overall in fewer than 30% of studies, of the blinded assessment of outcome in about 30%, of a power calculation, the y-axis has changed, in about 1% of studies, and a conflict of interest statement in about 10% of studies. So that's a problem, and the question is whether it's really just a problem largely for the neurosciences. You can see myocardial infarction performing reasonably well here. So next we went to PubMed. Now every publication in PubMed has got a unique PubMed ID, which is now a number between 1 and I think 26 million. So if you give me a random number between 1 and 26 million, I can give you a random publication. And we did that for 2,000 publications and found 147, about 7%, reporting in vivo experiments where randomization would have been appropriate. What we found was reporting of randomization in only 17%, of blinding in less than 3%, not one study reporting a power calculation, and about 10% reporting a conflict of interest statement. So it's not just neuroscience. It seems to be poor all over the piece. And when we look at the change over time, you can see there is an increase in the reporting. This is in quintiles of year publications. So the first quintile is from 1941 to 78, and the last is from 2008 to 12. And you can see there is an increase in the reporting of randomization. There is perhaps an increase in the reporting of blinding. But there's really quite a substantial increase in the reporting of conflicts of interest. And that's important because it implies that if we can encourage journals to introduce policies for randomization and blinding the same way that they did for conflict of interest reporting, we'll then be on to perhaps making some progress. You see there is no change in power calculations because they're normal throughout. So then we were told that if you take something at random from PubMed, of course it's going to be rubbish. And you've got to know the institution and the group and the team, so you've got to know it comes from a reliable source. Unfortunately, in the UK, we can identify what at least we think to be a reliable source because we have this thing called the Research Assessment Exercise, performed in 2008 and again in 2014. So we went to the 2008 Research Assessment Exercise and we identified 5 institutions which had been identified as making an outstanding contribution to the internationally excellent position of the UK in biomedical science, and where the panels had been particularly impressed by strengths within the basic neurosciences. And what we did for those 5 institutions, we went to PubMed and identified all of the publications we could find from those institutions describing experiments using non-human animals published in the 2 years after the RAE. And we found 1,173 publications from 5 leading institutions. This was published in PLOS Biology in October last year, so you can have a look at it if you like. Here we find reporting of randomization of about 15%, of blinding of less than 20%, of inclusion and exclusion criteria, one of the landest criteria for reporting, that you should be able to track every animal through the experiment and find out what happened to it. And power calculations at about 1%. And importantly, there are significant differences between institutions. These are color-coded rather than named to protect my future employment prospects. But you can see between those institutions significant differences that we were able to identify with 2 people working for about 3 months, in contrast to the very many person months that the RAE took to try and make similar judgments as to the rigor of research. So now we can actually do a comparison between leading UK institutions, the RAE, where for the years 2009-2010 reporting of randomization was less than 20%. If we go to the studies that we identified predominantly in the neurosciences and just focus down on 2009-2010, we find about 35% report randomization. So neuroscience is better than average animal research from leading UK institutions, but when we go to PubMed, for the same years, far fewer publications, but on average, publications in PubMed are selected at random, are of higher rigor, significantly higher rigor, than publications from the 5 leading UK institutions. So I do think that it's incumbent on institutions and others to do things about this. So journals, I think, need to think more about the ARRIVE reporting guidelines, and I know you'll hear more about this in the context of this conference. The Landis transparency guidelines, Nature have really stepped up to the plate with the change of their publication policy in May 2013, and we're involved with essentially an audit with them to ascertain what impact that's had, and we're also involved in a randomized controlled trial with another publisher to see whether specific editorial interventions can improve reporting. Funders really need to emphasize rigor and grant award, but as Rita Nagel and colleagues pointed out in Nature a few months ago now, the people who have a major role in this and who have yet to fulfill that role, I think, largely come from the institutions, who could do much more in terms of auditing the performance as an institution of scientists within their institution and providing CPD opportunities to try and allow people to improve that performance. Not in a punitive, aren't you awful, you've been very bad boys and girls, go away and do better, but in we're always interested, all of us, in improving the quality of the research that we do. How can we help you and us as an institution to do better quality research? I don't think anyone would have an argument with that. There's also a problem with how the data were analysed. Let me give an example from clinical research, the International Study of Infarct Survival 2, ISIS-2, recruiting when I was in short trousers as a junior doctor, which showed that aspirin improves outcome in myocardial infarction. But when you do a post-hoc analysis, there's a non-significant worsening of outcome for those born under Gemini or Libra. But what if those had been patients with migraine, where there was a non-significant outcome? Neurologists like me would have spent hours and hours and hours arguing in conference halls and bars about whether we should give aspirin to MI patients who also had migraine, when in fact it's a chance association. So let me show you that. These are Ontario has a very good health record system. And these are the odds ratios for hospitalisation for 5.3 million residents of Ontario by sign of birth. And you see, of course, that there are some associations here, significantly increased odds of admission for 24 of the 223 most common presentations. Scorpios having increased risk of lymphoid leukemia, Librans having increased rate of subarachnoid hemorrhage. I'm very glad I'm a Virgo, because I think I'm probably immune to excessive vomiting in pregnancy. But when you try and test this in a separate validation cohort, only 2 of these 23 come through. And then when you try and correct for multiple testing, none of these 24 conditions are actually found to be significantly associated. So there are real perils in testing multiple non-pre-specified hypotheses, and in many ways what happens in the lab can appear to an outsider looking in as being data collection followed by hypothesizing rather than the other way around. And there's also a problem with excess significance, which seems to suggest that we're only seeing the tip of an iceberg when we read a paper. So this is looking at over 4,000 experiments which we've collected in the context of systematic reviews across a number of different disease areas, and looking at how many of those we would expect to find positive findings on the basis of the underlying effect that was observed and the sort of variation that we were seeing. So this is work with Johnny Innes and his group, and we would expect of those 4,000 about 900 to be positive. In fact, we see about twice that. Highly significant observation. And what this suggests is that overall, over all of these diseases, it's not that this number is artificially high, it's that this number here, the 4,445, is artificially low. And this number should really be round about 8,000 to get 919 positive results. So a percentage suppression of experimental findings of about 47%. And are these multiple positive studies really plausible? Well, if we assume that a publication describes 6 experiments, each independent tests of an underlying hypothesis, and I assume that that underlying mechanistic hypothesis is correct, and that individual experiments using different cohorts of animals, different experimental setups, are each individually powered at 50%. And that's a, that's a relatively generous assumption. Most studies that we look at are powered at 30 to 40%. So the probability of at least one of those 6 experiments being positive is 98%. But the probability of all 6 being positive is only 2%, which perhaps goes some way to explaining, doesn't explain at all what we find when we read these papers. So if you go to PNAS or Science or wherever and read a paper that's got 6 independent tests of a hypothesis, every single test will be statistically significant. So this is what I think is happening. This is the fall off in the probability of getting n 1, 2, 3, 4, 5, 6 positive results out of 6 tests, falling from 100% down to 3%. What's actually happening, I think, is that the scientists are actually performing 12 studies, and they're only reporting those 4 or 5 or 6 that hit statistical significance and so are consistent with their hypothesis. So because they present a biased sample, those achieving significance, we are misled as the reader in terms of what actually happened. So journals, I think, could embark on pre-registration of studies. For instance, what's happening at Cortex, links to study protocols, for instance, in Open Science, and I know that some journals are exploring badges for this study's got data availability, this study's got a protocol available. Funders could pool published lay abstracts at the time of award and could try and develop between them A, a standard format for study protocols, and B, some clearinghouse websites. So instead of having to go to as many websites as there are funders to find out whether research has been done and organised. You could go to one place and get the answer. And scientists, and this is our practice, should publish their protocols, for instance, on Dryad or Figshare, and that'll give you a DOI and might even give you some citations. So incomplete publication is a real problem. This is the proportion of funded and completed research that is reported in years firstly from 1980 to 96 by different jurisdictions, then 97 to 2005 by different jurisdictions. And what you can see is that overall, on average, 50% of research is never published. And that's the same across countries, across research areas. The only funder that bucks this trend is the UK HTA, which does so by withholding 10% of grant award until the work is published. And there is evidence of publication bias. Here's our work in animal studies that actually Emily, who's moderating today, published 5 years ago now across stroke, experimental allergic encephalomyelitis and Alzheimer's disease models, where essentially we find that about 20% of studies are unpublished, and that leads to an inflation in efficacy of about a third. So when you correct for the presence of publication bias, efficacy falls by 32%. And it's interesting that the publication bias goes in different directions depending on the research field. So here studies in environmental toxicology, each dot's a study. To the right is having an increase in pup weight, to the left is having a reduction in pup birth weight. Reduction in pup birth weight is seen as being a reproductive toxic effect of the compound under test. And what you can see in red on the right-hand panel is those studies which imputation suggests were performed but not published. And when you correct then for publication bias, I showed you in animal disease models, we observe a 40% improvement in outcome. When we correct for publication bias, we see less improvement in that fall. But in tox models, where the outcome of interest is potential harm, we observe a 0.3 standardized mean difference harm. But when we correct for publication bias, there's actually more harm. So publication bias doesn't always go in the direction of understating effects. It can sometimes go in the direction of the interest of the person doing or funding the research. And it's important to do no harm. So some of you may be aware of the concern that there's been that neonatal anesthesia may impair cognitive development based largely on data from animal experiments. And when we looked at the animal experiments, we found 81 publications involving 3,000 animals, which we put together in systematic review. Of those measuring behavioural outcomes, such as here you see memory, only 16% blinded the assessment of memory outcome, 50% randomised, 5% did a power calculation, only 1% blinded the conduct of the experiment. We didn't meet our primary outcome because no study met all of those quality criteria. And for instance, in the Morris water maze, while overall there seemed to be a large and substantial effect, that effect was not present in studies at low risk of bias. And some of you will have seen the recent publication in The Lancet, a randomized trial of neurodevelopmental outcome at 2 years of age in a cohort of, or in a trial of children undergoing hernia surgery randomized to general or regional anesthesia with there being no effect. So animal research has the potential to do harm, not just in delaying the development of effective treatments, but also in encouraging parents, for instance, to withhold treatments from their children because they're concerned about a risk of harm which is ill-founded. So journals need to decide whether they're a journal of record or whether they're one of the less attractive journals, like we used to have the Sunday Sport or the Daily Mail. Funders, I think, should withhold 10% of grant pending publication. I'm happy for them to withhold 10% of my grant until I publish work from it. And I think we need to think more about new publication models. Institutions should encourage rapid publications anywhere, not simply vanity publishing in journals of the highest impact. If I've got a piece of work which I could get published tomorrow in an online open access index journal and people could start using it, or I could do another 3 experiments, tart it up, turn up the hypometer, and then maybe get it into Nature in 2 or 3 years' time, much better to get what I know out into the public domain now so other people can start validating or unvalidating or actually using the fruits of my research. And anything other than that adds delays to the research process. If the ethical conduct of research is premised on the basis that it will bring benefit to humans, then to introduce a 1 or 2 year delay while you make things fit for publication in Nature is, I think, not just unwise, but also unethical. And I think institutions also need to look at appointment panels, look at the work, not the packaging. And why look at the work and not the packaging? Well, going back to those 2,673 papers that we've got their risks of bias of, we also had the impact factors of the journals in which they were published. We were able then to look to see if there was any association between the two. So we divided the journals divided the publications by deciles of journal impact factor into 1 is the lowest 10% of journals by impact factor, 10 is the highest 10% by impact. You see a conflict of interest statement increases as impact factor increases, because journals with high impact factors tend to have a policy that you have to publish a conflict of interest statement. Sample size calculation is lousy everywhere. Blinding is pretty flat at 30% across the journal impact factors. And importantly, there is a statistically significant negative association between reporting of randomisation and journal impact factors. So the higher the impact factor, the more likely the work is to be at risk of bias. And so when my boss asks me, hey Malcolm, what have you published this year for our ref return? I say, great news. All of my work is published in low impact journals, which means it's highly likely to be true. And he looks slightly disappointed and shuffles out of the room. So studies can mislead if the sample size is wrong. Here is the systematic review that we did of the efficacy of TPA in thrombotic animal models of stroke, published in JCBFN. You can see there's almost 3,000 animals in total, but as you add animals, you get, so at the top here is the first meta-analysis of the first 2 studies, the next dot is the first 3 studies, the next dot is the next first 4 studies combined, and so on and so on. And as you add studies, there's a learning curve, and then there's another bit of a learning curve, but after you get to about 1,000 or 1,500 animals, you start not to get very much in the way of further incremental change. And we can look and see that NXY059 was taken to clinical trial on the basis of data from about 500 animals, so before this had flattened out, whereas for hypothermia, an intervention that we are testing in a clinical trial in Europe for stroke, the animal data is sufficient that we can be highly confident that we've got enough information that it's adequately describing what's happening in animals. Because small sample size and publication bias conspire together. So this was an observation from Hannah's EAE work that the larger the group size, here's more than 10 animals per group, the smaller the estimate of effect. So we set out to explore why that might be the case with some simulation studies. We assumed a complete publication bias, anything with p greater than 0.05 wasn't published, a true effect size of 10 with a standard deviation of 10, and we simulated 1,000 studies each for 5, 10, 15 and 20 animals per group. And if only p less than 0.05 gets published, only 30% of these studies here get published, whereas 86% of these studies here get published. And those of you who are awake will realize that this is simply a bootstrapped power calculation. But then when we take these 30% of studies that are published and combine them in meta-analysis, we find the estimate of effect size there is 60% higher at around 16% than the true effect size which we're trying to model. And even in studies which we would consider to be adequately powered, there's still an overstatement, bar a non-significant overstatement of effect in the overall estimate. So that's a problem of small sample sizes. How does that happen? Well, imagine 2 sets of studies. On the left here, of two studies each. On the left here, studies done with low statistical power. On the right here, studies done with high statistical power. Each of them testing an effect which comes in at about 25%. So if we take the average of each of these, then we get an overall estimate of efficacy of 25%. But that's not what happens, because in the underpowered studies, that study's missing. And so when we do the summary, that's the overall estimate that we get, and this overstatement in efficacy, in this case of 10%, is because of the suppression, the non-publication of non-significant, neutral, underpowered studies. So underpowered studies mislead. Statistical power is a function both of the number, the difference between groups, the variability, and what statistical threshold you choose. And efforts to increase power or reduce variance may also reduce generalizability. And that's an important caution, because the more we try and nail down our experiment so we've got very low variance, the more it'll be easier to say what happens in our experiment with greater precision, but the less those findings will be able to be generalized to other situations, to other animals, to other circumstances, to other laboratories. And this is my attempt to put into numbers Johnny and Ellis's paper, why most published research is false. In any study, you've got the statistical power and you've got the hypothesis testing. So if we take an alpha hypothesis of, if we take an alpha threshold of 0.05, that implies of those studies which are truly negative, 95% will be called negative and 5% will be called positive. If we've got a power, and I've taken power here of 40%, then if our biological effect is truly there, then 40% of the studies will find it and 60% of the studies won't. What people miss when they think about significance testing is that it's critical to know what the proportion of potential studies are that are truly positive or truly negative. Because what you're interested in isn't the result of your p-test, which is if this was negative, only 5% of studies would find this answer. What you're interested in is, if I've got a positive result, how likely it is that that positive result is a true positive. So here, we're assuming that 20% of hypotheses are correct. We're assuming we're powered at 40%, and we take a p-value of less than 0.05 as a threshold. So of those, 20% of studies will be positive. Of those 20% of studies that are positive, 40%, and 40% of 20% is 8%, 8% will test positive, 12% will test negative. Of the 80% of studies which are truly negative, 95%, which is 76%, will test negative, but 4% will test positive. So overall, we'll get 12% of studies will test positive, of which 8% will be truly positive and 4% will be truly negative. So the positive predictive value of our test is 67%, not 95%. And that's not well understood. Let's go hunting. If 1% of hypotheses are correct, then with power at 40% of p and p of less than 0.05, the positive predictive value, if you work it out, is 7%. Even if you take p of less than 0.05. So how do you get to something closer to what you expect? Well, if 20% of hypotheses are correct, I don't think that's an unreasonable starting point. If power is at 80% and you take a p of less than 0.01, then you get a positive predictive value of 95%. So I would argue that we need to change the way that we think about doing our experiment, that instead of taking power of round about 40 or 50% and a p-value of 0.05, we should take power at 80% and a p-value of 0.01. So if you look at the combination of p-threshold and statistical power required to give that positive predictive value of 95%, where the prevalence of truly positive studies is 1 in 5, then you can see that this line, when you've got absolute statistical power, your p-threshold is about 0.013, and if you're powered at 40%, then the p-value that you would have to take threshold would be 0.005. And that powered at 80%, a p-threshold of 0.01 is, I think, a happy medium of the achievable versus the desirable. So improvement strategies funded to require real power calculations and to provide expertise in experimental design. And some of you may know that the UK NC3Rs has recently launched the Experimental Design Assistant, which I would recommend to you. And I think regulators should end the tyranny of reduction. You know, there are many reasons why experiments are too small, but one of them is that so people can write in their ethics application, we have reduced the number of animals to the minimum required, and that also happens to minimize the time I need to spend in the experiment, and it also minimizes the cost associated with the experiment. And nobody calls out a study for being underpowered, and we should call out studies for being underpowered. Institutions should provide in-house expertise in statistical design, and as scientists we should cut our cloth according to our coat. And what I mean by that is that if we don't have enough experimental subjects to answer the question that we want to pose with sufficient statistical power, we should ask a different question, because there's no merit in doing underpowered experiments, no merit whatsoever. And there are other cautions. HARKing, I talked a little bit about earlier on, hypothesizing after results are known, where you collect the data, you run a number of different outcome measures, and then you try and put together a hypothesis that fits the one that's met a p-threshold of 0.05 by chance. Flexibility in data analysis, where you perhaps have an experiment where it's possible to measure the outcome in different ways. So in our Alzheimer's work in the Morris Water maze, in 53 publications, Kieran found 59 different statistical analysis approaches to measuring the outcome in the Morris Water maze. And you can imagine that people don't use all of these different outcomes just because they've got a personal favourite, but rather they'll report the ways of measuring the outcome that happen to fit the hypothesis. And you could get around a lot of these problems, I think, by the introduction of protocols, which are population, defined, hypothesis, intervention, sample size calculation, primary outcome measure, and statistical analysis plan. And you could set that out just on one side of A4 with 10 words under each. Lock that away on your institutional website, and when you submit your paper for publication, give the link to the reviewers. And when you publish your paper, give the link to your readers so they know that you actually did what you set out to do. A good test of a hypothesis, of course, will give you a clear answer. The hypothesis will either be confirmed in green or refuted. But the trouble is that many of our hypotheses allow us, because of wooliness in the way in which they're characterized, to be very flexible, I suppose is a word, in the way in which the results are interpreted. And remember what Karl Popper said, some genuinely testable theories, when found to be false, are still upheld by their admirers. For example, by introducing ad hoc, some auxiliary assumption, or by reinterpreting the theory ad hoc in such a way that it escapes refutation. Such a procedure is always possible, but it rescues the theory from refutation only at the price of destroying, or at least lowering, its scientific status. And it's much better to spend more time getting a hypothesis right and being a difficult test of your hypothesis, rather than having something quite woolly which you know is going to give you a bit of wriggle room if you don't get the answer that you like. Animal research can go wrong if people use the wrong model. So here's hypertension and stroke, NXY059, I talked about this briefly earlier on. The overall estimate of efficacy in the vertical grey bar, but in hypertensive animals here, much reduced efficacy. 7% of animal studies were in animals with hypertension, 77% of patients in the neutral SAINT-2 study had hypertension. Here's clot-busting treatment. Here's the treatment that works, TPA. 9% of animal studies we've used animals with hypertension doesn't do anything in animals with hypertension, but fortunately, and I think it probably was fortunate, the pivotal NIM study of TPA and stroke specifically excluded patients with a history of hypertension. So the TPA research was done in humans, which were most like the animals in which efficacy had been shown. And here you can see some validation of animal models. This is again TPA and clot-busting treatments for stroke. This is the decline in efficacy in 90-minute epochs from the time of occlusion in animal studies on the left and in clinical studies on the right. Each involve about 3,000 subjects, so they're fairly robust estimates. Those error bars are the 95% confidence intervals rather than standard deviations or standard errors. And you can see that those lines, while they don't sit completely exactly on top of one another, tell us the same general story, that over the first 4 to 6 hours after stroke, there's a decline in efficacy to not very much efficacy being left. So scientists, I think, need to develop better validation of their animal models, perhaps done through targeted experiments. And ALS.org did this for motor neuron disease, for ALS, with remarkable results. Systematic reviews of hundreds of experiments, as we have done, comparing models and different outcome measures to understand whether the animal model does have implausibilities within it, implausibilities in the data that are shown that mean that it is of limited or reduced value. And I think also once we've got our validated animal studies, we need to think about different ways of doing those pivotal proof of concept preclinical trials before going into human clinical trial. and we've advocated and done the preparatory work to allow multi-centre animal studies to be done in the same way that we do multi-centre human clinical trials. So we can improve the chance of translational success if we systematically review the available data, conduct further in vivo experiments if indicated, design our clinical trials according to what the animal data actually say, not what we would like them to say, and develop tools, and we're involved with a number of groups doing this, to allow rapid, living, systematic reviews of animal data so that clinical trial decisions can be informed by the very latest information from the laboratory, not information that's perhaps 1 or 2 years old. And if you look at the people that I've been criticising for the last 50 minutes, they're the funders and the journals and the regulators and the institutions. But at the heart of each of these are the peers, because the peers who review for the funders, who review for the journals, who sit on regulatory panels, who run our institutions. So this isn't a problem that's out there that we need to get other people to fix for us. It's a problem that we can start to fix every day in our working life, and we should all take the pledge and start to do so. Science is not, as currently conceived, a profession. We don't have a code of professional ethics, we don't have a regulatory body, we don't have continuing professional development. Even professional football players, soccer players, for those of you in the States, have a code of professional ethics. They have to do 16 hours a year, where presumably, even in Uruguay, they're told that they're not supposed to bite their opponents on the shoulder. And if they don't do their CPD, they're not allowed to play. And scientists don't have that, and we should. And we should have a code of professional ethics, and we should have robust and systematized tools for continuing professional development, perhaps enforced or reinforced or supported by annual programs of appraisal, done not for accountability, how many grants have you got, how many papers have you published, but appraisal by a peer outside the immediate research team to try and explore with the scientist what they might be able to develop in terms of their scientific toolkit to improve the research which they do in the future. So my key messages today: in vivo studies which do not report simple measures to avoid bias give larger estimates of treatment effects, and unfortunately most in vivo studies do not report simple measures to reduce bias. Publication and selective outcome reporting biases are important and prevalent. One cannot assume rigor even in journals where one would think that you could assume rigor. We've only been able to find this stuff out by studying large numbers of studies, and I would encourage you either to do that yourself or to support others who do. And crucially, any experimental design can be subverted. What's important is learning how to recognize when that's happened. So how big a hole does this make in published research as relates to animals? Well, if this is the totality of published research, you lose about 20% because of risk of bias, 20% of the value. I would argue that you lose about 20% from publication bias from the 20% of studies that aren't published. They're kind of clearly lost. And I think you lose about another 20% from underpowered experiments. So overall, one could argue that 60% of animal research is not as efficient, is not as useful as it might otherwise be, because of these three very simple reasons that we could all do something about fixing about tomorrow. I'd like to thank my collaborators, particularly Emily, but the whole extended CAMARADES team, our funders. If you'd be interested in doing a systematic review, we're here to help. You could send me an email, I'd be delighted to reply. And I think now we can have a question and answer session, but thank you for your attention.
Thank you, Malcolm. That was interesting. After all these years, I still learn new things, which is good. So as Malcolm said, we're about to get started with our question and answer session. I'd like to remind the audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is from Sherry Gross from UC Davis. She asks, will you please clarify the need for replicate studies if power analyses have been used to determine the number of animals needed for statistically valid results, in contrast to having reproducible studies that can be achieved by another lab at another institution?
Yeah, great question, Sherry. So there's, if you want it to be Bayesian, you start off with a prior, and you do your study, and then you apply your Bayes factor, and then you get a posterior probability that what you're seeing is true. So one of the reasons for replication would be if your prior probability is so low and your prior probability is so low that even with an adequately powered experiment, you're still getting a posterior probability only up to 50 or 60%. You might want yourself to do another study to get that posterior probability up to 90, 95%. Certainly in clinical trials, the FDA would require 2 or potentially 3 clinical trials for a stroke drug, because such is the graveyard of stroke drugs that even with a trial positive at 5%, the posterior probability that that drug actually works is only around about 38%. So that's the first reason. But actually, I think there's a much more important reason, and that's about the generalizability of findings. So there was some great work that was done a long time ago, published in Science by a guy named Crabb, I think, where they looked at the replication of behavioral findings in 8 different groups of animals for 8 different outcome measures across 3 different labs in Portland, Albany, and I think Calgary, but I may have got that wrong. And for instance, in measures of anxiety, they had a 5-HT knockout animal, and that animal compared with its wild type. In one lab, the knockout seemed to increase anxiety. In another lab, it significantly reduced anxiety. In a third lab, it had no effect. And you were getting absolutely tangibly different results using the same animals, using the same protocols, just by different tests. And so there's an issue about generalizability here. Hanno Werbel and colleagues has done some very interesting work about how, and you can think particularly in the question in the issue of developing a drug for stroke, where you're trying to breach that species barrier. If you've already been able to breach a species barrier from mouse to rat to rabbit to cat, for instance, then one might assume that the probability of being able to breach that species barrier to humans would also be higher. So the more homogeneous the circumstances of testing, the more precise the estimate, but the less generalizable. The more heterogeneous the circumstances of testing, the more animals you need, but I think the more robust the finding. And so we are starting to try and articulate a model whereby early experimental discovery work is done in highly homogenized single-site experiments to try and work out exactly whether there's a signal there. But before taking it to clinical trial, one embarks on the polar opposite type of experiment with lots and lots of heterogeneity and lots of different centers, on the basis that a drug that shows efficacy across that wide range of circumstances is much more likely to generalize to human clinical trial. And that's one of the main drivers or principles behind the multi-center approach, multi-parts approach to preclinical multi-center clinical trials for animals, because we think it will give us much more robust answers.
Okay, thank you. We'll just give it a couple more seconds if we get any more questions. Okay, I think we don't have any more questions, and once again I'd like to thank Malcolm for his presentations. Do you have any final comments that you want to
No, just thank you all 215 of you for turning 214 now, for turning up, and I hope the rest of your day is interesting and successful. Thank you very much.
Thanks, Malcolm. So today's webcast will be available for on-demand viewing 6 months from today, I believe. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you again for attending, and goodbye.